Hey everyone, today I am going to talk about glycolysis. Glycolysis. It is a nearly universal 10-step metabolic pathway for producing ATP by the oxidation of glucose. In this process, two molecules of ATP are invested to activate glucose, but the products of the pathway include 4-ATP as well as NADH and the triose pyruvate, which can be metabolized further in other pathways. Step 1. Glucose is phosphorylated by ATP to form a sugar phosphate. The negative charge of the phosphate prevents passage of the sugar phosphate through the plasma membrane, trapping glucose inside the cell. Here, glucose in the presence of hexokinase and an ATP forms glucose 6-phosphate. In the step 2, a readily reversible rearrangement of the chemical structure, that is isomerization, moves the carbonyl oxygen from carbon 1 to carbon 2, forming a ketose from an aldose sugar. Glucose 6-phosphate in the presence of the phosphoglucose isomerase enzyme forms fructose 6-phosphate. In step 3, the new hydroxyl group on carbon 1 is phosphorylated by ATP in preparation for the formation of two 3-carbon sugar phosphates. The entry of sugars into glycolysis is controlled at this step through regulation of the enzyme phosphofructokinase. Fructose 6-phosphate in the presence of the enzyme phosphofructokinase and an ATP molecule forms fructose 1,6-biphosphate and release 1 ADP molecule. In step 4, the 6-carbon sugar is cleaved to produce two 3-carbon molecules. Only the glyceraldehyde 3-phosphate can proceed immediately through glycolysis. Fructose 1,6-phosphate in the presence of enzyme aldolase and an ATP molecule releasing ADP and forming glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, which can be interchanged to glyceraldehyde 3-phosphate and as when required. In step 5, the other product of step 4, dihydroxyacetone phosphate, is isomerized to form glyceraldehyde 3-phosphate. Dihydroxyacetone phosphate in the presence of the enzyme triose phosphate isomerase forms isomerizes to glyceraldehyde 3-phosphate. In step 6, the two molecules of glyceraldehyde 3-phosphate are oxidized. The energy generation phase of glycolysis begins as NADH and a new high energy anhydride linkage to phosphate are formed. Glyceraldehyde 3-phosphate in the presence of enzyme glyceraldehyde 3-phosphate dehydrogenase forms 1,3-biphosphoglycerate and 2-NAD plus forms 2-NADH plus 2H plus. Step 7, the transfer of ADP of the high energy phosphate group that was generated in step 6 forms ATP. 1,3-biphosphoglycerate forms 3-phosphoglycerate in the presence of the enzyme phosphoglycerate kinase and 2-ADP forms 2-ATP. In step 8, the remaining phosphate ester linkage in 3-phosphoglycerate, which are relatively low free energy of hydrolysis, is moved from carbon 3 to carbon 2 to form 2-phosphoglycerate. 3-phosphoglycerate in the presence of enzyme phosphoglyceromutase forms 2-phosphoglycerate. In step 9, the removal of water from 2-phosphoglycerate creates a high energy enol phosphate linkage. 2-phosphoglycerate in the presence of the enzyme enolase forms phosphoenol pyruvate and two molecules of water are released. In step 10, the transfer to ADP of the high energy phosphate group that was generated in step 9 forms ATP and thus completing glycolysis. Phosphoenol pyruvate in the presence of enzyme pyruvate kinase forms pyruvate and two molecules of ATP. Summary of glycolysis. In this glycolysis process, one molecule of glucose forms fructose 1,6 biphosphate, and it uses one ATP molecule in step 1 and one ATP molecule in step 3. These steps are known as energy investment steps. Then fructose 1,6 biphosphate forms two molecules of glyceraldehyde 3-phosphate in step 5. These steps are known as cleavage of 6 carbon sugar to 3 carbon sugars. Then glyceraldehyde 3-phosphate forms two molecules of pyruvate, and in between, in step 6, it releases two molecules of NADH. In step 7 and step 10, it releases two molecules of ATP each, and thus these steps are known as energy generation steps, and the final product of the glycolysis is two molecules of pyruvate.